In this video, we will explore the development of the integumentary system, including the skin, hair, sweat glands, and associated structures. We will discuss the processes of self-renewal and differentiation of epidermal stem cells, formation of sweat glands, folliculogenesis, metalloproteinase function in skin development, the role of Langerhans cells, keratinocyte differentiation, dermal papilla cells and hair follicle morphogenesis, neural crest cells and melanocyte differentiation, wound healing, Eccrine glands, hypodermis development, and genetic regulation of skin pigmentation. Let's begin our journey into embryonic development and the integumentary system. In the video, we will explore the fascinating world of epidermal stem cells, which are responsible for the continuous renewal of the outermost layer of our skin. We will delve into the mechanisms that govern their self-renewal and differentiation, including the roles of signaling pathways like Notch, WNT, and BMP, as well as transcription factors like P63, SOX2, and KLF4. By understanding these mechanisms, we can potentially develop new therapies for regenerative medicine and skin disorders. Sweat glands are crucial for regulating body temperature and eliminating waste through sweat. Eccrine and apocrine sweat glands are formed through complex signaling pathways involving WNT, FGF, and Notch signaling. Transcription factors like FOXC1, HOXC13, and FAC1 are also important for sweat gland development. Understanding these processes can help develop treatments for sweat gland disorders. Hair follicles are complex structures involved in hair production and growth. Their development, called folliculogenesis, requires communication between different cell types. It starts with the formation of placodes, which give rise to follicles. This process is regulated by various signaling pathways and transcription factors. Understanding folliculogenesis can help develop treatments for hair loss. Matrix metalloproteinases, MMPs, are enzymes crucial for skin development and healing. They remodel and degrade the extracellular matrix, allowing tissue remodeling and cell migration. Dysregulation of MMPs can lead to skin disorders, highlighting their potential as therapeutic targets. Langerhans cells, found in the epidermis, are vital for skin immunity. They originate from bone marrow, migrate with chemokines, and capture antigens. They then move to lymph nodes, activating immune responses and preventing excessive reactions. Dysfunction can lead to skin disorders. Understanding Langerhans cells unveils the intricate connection between skin and immunity, leading to potential therapies. In the section on keratinocyte differentiation, we explore the intricate signaling cascades and regulatory factors involved in the formation of the epidermis. Key players include Notch and WNT signaling, keratin gene expression, and factors like EGF and retinoic acid. Understanding these mechanisms is crucial for studying skin diseases and promoting epidermal regeneration. Dermal papilla cells are important for hair follicle development and growth. They communicate with epithelial cells through different signaling factors to regulate the hair growth cycle. Transplanting these cells has potential for hair regeneration in alopecia cases. Further research is needed in this field. During embryonic development, neural crest cells give rise to melanocyte, which produce pigment. These cells migrate to the epidermis, and their differentiation, migration, and distribution are regulated by various signaling factors. The complex interplay of factors like SCF/c-KIT, endothelin-1/EDNRB, and WNT signaling controls the growth and movement of melanocyte precursors. The distribution of melanocyte in the epidermis is determined by paracrine signaling, cell-cell interactions, and interactions with the surrounding environment. Disorders in melanocyte development and distribution can lead to pigmentation disorders such as albinism and vitiligo. Understanding the biology of melanocyte and neural crest cells could potentially help in the treatment of skin disorders like pigmentary disorders and skin cancer. Summarize in 33 words. Wound healing in the integumentary system is a complex process involving immune cells, fibroblasts, and epithelial cells. Molecular factors like TGF-beta, FGF, and MMPs play important roles. Understanding this process is crucial for developing effective therapies for wound closure and tissue regeneration. Eccrine sweat glands are formed from the epidermal placode, invaginating into the dermis to form the sweat gland primordium. Molecular factors like WNT, BMP, Notch signaling, and transcription factors like PAX6, SOX2, and RUNX1 regulate gland development and differentiation. Proper sweat secretion is essential for thermoregulation and electrolyte balance. The hypodermis, also called subcutaneous tissue, consists mostly of adipocytes involved in energy storage and insulation. During development, adipocytes differentiate from mesenchymal progenitor cells in the dermal layer. Signaling pathways like progamma, WNT, and hedgehog and transcription factors like SEB-alpha, SEB-beta, and SREB1C regulate this process. Paracrine factors from neighboring cells play a role too. Understanding hypodermis development is important for understanding metabolic disorders. The color of our skin is determined by a combination of our genes and the environment. Key genes, such as MC1R, TYR, and OCA, regulate melanocyte function and melanin synthesis, which are responsible for skin color. Mutations in these genes can cause pigmentary disorders. Transcription factors, epigenetic modifications, and signaling pathways like MOPK and CAMP are involved in regulating skin pigmentation. Admixture and natural selection also contribute to the diversity of skin color in different populations. Understanding these genetic mechanisms is important for dermatology and treating pigmentation-related disorders. In conclusion, the development of the integumentary system is a fascinating and intricate process involving various cellular and molecular events. Understanding these processes can lead to advancements in regenerative medicine and dermatology.